So welcome. Thank you for being here. It's great to see such a crowd here. I'm Diane Lipscombe.
My name is Bjorn Sandstede. On behalf of both of us, I would like to welcome you. This event is part of the university's Building on Distinction lecture series. It marks the launch 5 years ago of the strategic plan that Brown has now in place. I think the purpose of this lecture series is to highlight exceptional research that's done at the interface of different disciplines, and that showcases this research in this series. So it's particularly timely to have this at this time. In a month's time, the Carney Institute and the Data Science Initiative are going to move into the same space, the former Brown office building, 164 Angel Street, which we are both very, very excited about. We're going to be on the 3rd and 4th floor. We have a lot of shared space, and we envision a lot of interactions between the two units and a lot of exciting research coming out of this.
That's above the bookstore, 164 Angel. I've often thought, what would be the most wonderful place to have my office, and it would be above a bookstore, and so we're going to be up there. The machines and everything will be gone soon, and you'll be able to walk back and forth along Thayer Street. So as Bjorn said, I think it's really exciting for us. When I first spoke to Rick, about moving out of Stimson, which is the home of the Institute right now, next to the Athletic Center, and we were talking, and he had mentioned that there was the possibility that this space would be available. And we brainstormed a bit, and we thought how brilliant it would be to have the Data Science Initiative and Brain Science kind of located in the same building. And like many things that Rick has done over the last few years, he made it happen. So thank you. It was just a brilliant idea to bring kind of an academic focus into that fantastic kind of central place on campus. And so we're indebted to you for really making this happen. I do also want to mention a couple of other people, and I don't know if I see them here, but Leah Van Wey has been brilliant in helping us through the process of the building, holding our hands, saying it's going to be okay, it's going to be wonderful, and really producing and helping us to have this absolutely beautiful space, which you will all be invited to. So finally, I just also wanted to mention, as Bjorn said, that we're really going to work as a unit. The point of being together is to really enhance collaborations. You'll hear today of the existing collaborations that we have across these two disciplines, and I think you'll find their research incredibly exciting, but that's just the beginning. And we really think that being located in the same space is just going to continue to enhance and bring new innovative and creative research onto campus. And finally, I just also wanted to say that it's not just data science and brain science, Carney Institute going into the building, but also CCMB, Center for Computational Molecular Biology, run by Sohini Ramachandran, and also the Annenberg Institute. So it's going to be a data-intensive, data-heavy, data-driven research and science going on there. So thank you all for being here. Do you want to say anything else? So I now wanted to just bring up, we'll get the show going, and introduce Cornelia Dean. You'll be the moderator, and we're absolutely thrilled that you agreed to do this. You barely need introductions, but you will see everybody is listed in here with a great description. But Cornelia is a visiting professor at Brown, a science writer, and the former science editor for the New York Times. Cornelia, thanks.
Thank you. So we are here today to learn about 3 fascinating areas of research, all of them combining cutting-edge investigations in neuroscience with advances in data science. To me, these projects embody the potentially enormous advantages we can derive from true interdisciplinary research. There are still plenty of people who deride interdisciplinary research as resulting in collaborations that do not dive deeply enough into any of their component parts. As you will see from the presentations you see in here today, that is not necessarily the case. Today we will hear from Matthew Harrison of Applied Mathematics, Stephanie Jones of Neuroscience, and Dima Amso of Cognitive, Linguistic, and Psychological Sciences on 3 projects that reach across disciplinary lines to produce research with potential to change lives. In our format, each of them will speak for about 10 minutes, and then we will have a discussion in which speakers will take questions from the audience. We ask you to hold your questions until the discussion period. In the interest of time, I will skip the usual descriptions of people's academic histories and so on, all of which are in your program, and just get straight to the heart of the matter. So our first speaker is Matthew Harrison, Associate Professor of Applied Mathematics here at Brown. Among other things, his research focuses on applications relating to the brain-computer interface and technology that can enable people with paralysis to communicate and do other chores. So here is Matthew.
And welcome everyone. So today I want to tell you a little bit about one of my collaborations, and it's my collaboration with the BrainGate group here at Brown. The BrainGate group involves an enormous number of people who made possible the research that I'm going to tell you about, and I just collaborate with them. So they develop brain-computer interface technology to improve the lives of people who are paralyzed or who have lost limbs. And so one of the types of technology that they develop is they work on a sensor that can be implanted on the surface of the brain under the skull. And the pattern of brain activity recorded by this sensor can allow people to control devices with their thoughts. So here's an example of a paralyzed woman using the device to type on a computer. You can see the device attached up to the top of her head. She's unable to move any of her limbs, but she is able to control a computer cursor with her thoughts using this BrainGate technology. And so she was asked the question, how do you encourage your sons to practice music? And she's typing, when they started their lessons, they were almost 3, and they took to playing violin and cello very fast, and I had a sticker system, and so on. So in this next video, you'll see a man with paralysis controlling an experimental artificial hand for the first time. So you can see the hand there. It's connected to the BrainGate technology. And even though you're going to hear him saying open and close, the artificial hand is not voice activated. It's controlled with his thoughts using the BrainGate technology.
Open. Not there, right? Open.
So here's an example of the type of signal that the BrainGate sensor records. Every now and then you'll see a big sort of spike of electrical activity, and that's a brain cell firing close to the sensor. And the sensor simultaneously records dozens or 100 of signals like this. And a single participant generates an entire hard drive's worth of data every day, and that's only increasing as the technology improves. And so at the heart of the BrainGate technology are mathematical and statistical algorithms that translate these brain signals into commands that a computer can understand. Now, of course, the brain is a giant mystery. Nobody understands how it works. And the computer that's receiving these signals from the sensor has no idea what they mean or how to use them, at least at first. So in this next video, you're going to watch in real time as the computer learns how to interpret the brain signals recorded by the BrainGate sensor. And the person, a person with paralysis, is trying to move the cursor to the highlighted dot. You'll see the cursor in a minute. That's what they're instructed to think about when they play this game. So you'll see they're trying to move it. There comes the cursor. It's not really working. And so the computer begins with zero knowledge. And consequently, the algorithm doesn't allow the participant to control the cursor in a meaningful way. But over time, as this game goes on, the computer discovers statistical patterns in the brain data that allow it to predict how the user is trying to move the cursor. So the person is trying to move it. After a while, it times out and goes to a different target. And all the while, the sensor is recording brain data, and it's presuming that they're trying to move to the right location. So that allows it to relate brain data to movement intention. And slowly you'll see the algorithm improve, and the person can begin to control this game. So my collaborations with BrainGate have focused on how to improve the statistical learning algorithms at the heart of this technology. And this video is showing one of the algorithms that I helped develop, along with doctoral students in neuroscience and applied math and biomedical engineering. And so the person's a minute into playing this game, and after a minute or two, they have pretty good control already, and it will continue to improve. So once they've calibrated using this game and the algorithm understands how to interpret their brain data, then they can move on to more interesting tasks like typing on a computer. So let me just spend a little bit of time trying to give you a sense of how the algorithm learns to translate brain signals. So the sensor is collecting brain data. But it can also guess what the person was trying to do, so this is symbolizing like a movement direction or an intention, because it knows that the person is trying to play this simple game of moving the cursor to the highlighted dot. And in the next instant of time, it gets new brain data and a new movement intention. And this goes on. And over time, it starts to build a dictionary, so to speak, of brain data movement intention pairs. Now, when it gets new brain data, once it has this dictionary, and let's say the person is typing, it's not playing the game anymore, so it doesn't know where the person's trying to go, then what it can do is it goes back to the dictionary that it's built, and it tries to situate this new brain data somewhere in the pattern of all the brain data it's collected to fit it into that dictionary. And then once it's done that, it can use it to predict what direction the person was intending to go. And then it moves the cursor a little bit in that direction, or moves the robot arm. And of course, underneath the hood of all this, there's a lot of math, and applied math, and data science, and a lot of it to make it work fast enough to be of use in this brain-computer interface technology. And I think really a lot of continuing opportunity for applied mathematicians and data scientists to further improve the algorithms that lie at the heart of this truly revolutionary technology that's being developed by the BrainGate group right here at Brown. So, thank you.
So our next speaker is Stephanie Jones, Associate Professor of Neuroscience. Among other things, her work integrates brain imaging and computational neuroscience to study how the brain works, both in people, healthy people, and in people with problems with the brain.
So thank you, and again, welcome everybody. Very glad to have the opportunity to describe to the community a new software tool that we've been developing to study the human brain signal. And you'll see when I describe the development and applications of this tool that it integrates many aspects of both data science and neuroscience. So I want to begin with the question that we were trying to address with this new tool. And as Matt just showed you, we can get really powerful and predictive information from the human brain when we record directly from the surface of the human brain. But these are highly invasive procedures, and they're only performed in select patients that have a very specific need. And so what I'm going to do is I'm going to move the discussion to signals that we can record non-invasively outside of the head. And the question that we asked is, can we develop a technique to interpret these non-invasive human brain signals in both healthy behaviors and in pathology? Sorry, my slide got cut here. That must be something about transferring to this computer, so we'll just go with it. But that says, and pathology. Okay, so one of the most powerful ways that we can record human brain signals non-invasively is with a technique called EEG, or electroencephalography. Now, EEG is very powerful because it measures the electrical signals of the brain with very fine temporal resolution on the order of milliseconds, similar to what you saw in the data that Matt showed. And it's very easily to use. And it can be done in awake behaving children, including infants and babies. So I'm showing you here an example of a well-known signature of activity that's recorded with EEG. When you put an electrode over the part of the brain that represents the visual cortex, and you close your eyes, you see this large amplitude rhythm that emerges. And when you close your eyes, and when you open your eyes, that amplitude of that rhythm dampens. Now, EEG is an old technology. It's been around for almost a century. The first human EEG recordings were done in the early 1900s by a psychiatrist named Hans Berger. And at that time, they didn't have computers, and so they had to store the data as drawings on paper. Now here we are in the 21st century. We have massive computer power. We have the ability to store vast amounts of data. We have now open science that allows us to share this data worldwide. We have all these new machine learning and other algorithms that are allowing us to classify different brain states. And we've reached a time where we have these vast data streams and data mining capabilities. And now these EEG signals are associated with almost every healthy and pathological brain state there is because they're so easy to obtain. But there's a big downside to EEG. And what we're recording is considered this macro scale signal. And it's still very difficult to infer how are these signals generated by the underlying neurons and neural networks within the brain. And this network level understanding is really critical if we want to know why these signals correlate with information processing, or if we want to be able to target treatments when they're disrupted in neuropathologies with either pharmacology or other therapies. Now, at the same time, over the past century, we've also had a revolution in our ability to study signals within the brain with electrophysiological, imaging, and genetic tools in animal models, and sometimes in humans, as you saw. So we're now having this unprecedented level of cellular and network information. And again, these signals are correlated with nearly all healthy and pathological neural dynamics. But what we need is a translator that can connect these macro scale signals that we can record outside of the head and these vast data streams we're collecting in humans with the underlying network level information that's creating these signals. Now, this is the perfect job for a technique called computational neuromodeling. This is where we simulate the electrical activity of the neurons on the computer. And we can have specificity both at this cellular and network level and at this macro scale recording level. And this has been the primary focus of my research program. It again builds from a long history of science. Back in the 1950s, Alan Hodgkin and Andrew Huxley established that you could write the electrical activity of a neuron with a mathematical equation. Now, back then, they had to solve these equations by hand, and so you could only study really reduced representations of neural circuitry. Now we have computational power that lets us simulate large-scale networks in large portions of the brain. And so what we've been doing is we've been taking advantage of this computational power in developing a large-scale network model that we've turned into a user-friendly software tool for clinicians and researchers to start to develop and test hypotheses on the cellular and network origin of their data. So to give you a sense of how this, we're calling it human neocortical neurosolver, which is quite a mouthful. But that's because the underlying neural model that we're simulating represents the outer layers of the brain called the neocortex. And it's these outer layers that contribute to these EEG signals that we can record outside of the head. But I'm just going to refer to it as HNN for simplicity. So to give you a sense of how this is used in practice, I'm going to describe an application of HNN where it was applied by Tal Knet in a group at Mass General Hospital to interpret abnormal brain dynamics in children that had autism spectrum disorder as compared to typically developing children. And what Tal and her group did is they had these children come in and they gave them a 25 hertz vibrotactile stimulus to the middle and index finger. And they recorded their brain activity. And what I'm showing you here is they applied what's called an inverse solution technique to the data to isolate the contribution to the signal from the hand representation in the brain. It's in an area called the primary somatosensory cortex, or S1. And so what I'm showing you here is the electrical activity from this hand area of the brain. And what they saw is that this is during this 25 hertz vibrotactile stimulation. During this steady state period, there were some subtle differences in the typically developing versus autistic children. They performed another analysis where they looked in the frequency domain. The frequency domain measures how fast do these signals oscillate in time. And here they saw clear differences in these two groups, such that the autistic subjects had a stronger response at the stimulation frequency and a weaker response at this higher frequency around 50 hertz. And they had a hypothesis about, well, what might be the difference in the brain circuits of the children with autism compared to these typically developing children, based on prior research. And what they hypothesized was, well, perhaps in these children that have these tactile sensitivities, there's an increase in the communication from the periphery into this part of the brain. We call that a feed-forward pathway. And there's a decrease in communication within the brain through these pathways that we call feedback pathways. And so what they wanted to do is to take our model, simulate these differences in the neural network, and see, could this simulation account for the differences that they saw in their data? And so I'm going to briefly walk you through how you might do that in the model. So here again is their data. When you start the HNN software, this is the graphical user interface that appears on your screen. And what's shown here is a schematic representation of the underlying model. And it represents, sorry, was that me? That was you guys. It represents a patch of cortex. And so we're simulating this little patch of cortex in the brain. It also has cellular and network level information, including the local connectivity, and these pathways of information flow into the local part of cortex through these feed-forward and feedback projection pathways. And so what they could do is they could simulate this vibrotactile input by giving activation through these feed-forward and feedback pathways, which I'm showing you right here is an envelope of the activation curves that drive this local network. And this is the responding electrical signal that the computer calculates. And they can compare that to their data. And what they did was they tuned the model. All the parameters in the model are tunable. They tuned it so that they had an accurate representation of the typically developing data. And then they wanted to test their hypothesis. If they increase the strength of the feed-forward input and decrease the strength of the feedback input, will that accurately account for the differences they're seeing in their data? And so they go into our model, and they change the strength of the feed-forward input and make it effectively stronger. And they change the strength of the feedback and make it weaker, and then re-simulate the electrical output of this network. And what they saw was they did see similar differences, but in particular, when they went into the frequency domain, they found that this was able to accurately reproduce these signature differences that they saw, lending support to their hypothesis that what's happening in these autistic children is perhaps there's an increase in the feed forward and decrease in the feedback. Now, because this is a model, you can go in and change any parameters you want. And so they could test an alternative hypothesis. Perhaps in these autistic children, there's a degradation of the communication across the brain. And so these signals don't come in very clearly, but they're more dispersed. And so we can simulate them to come in more dispersed. And here's the envelope of the input, and you can see now that it's more broad and dispersed. Now the electrical signature is very different. And when they look in the frequency domain, it looks nothing like the data that they collected. And so they can rule out this hypothesis. And so in this way, you can generate all sorts of families of responses and compare them to your data to see which is the best fit to account for the differences in the brain of these autistic children. Now, one thing I'm not showing you is you can also go in and look at the circuit and cellular level details. So you can look at the spiking activity of the cells. And this gives you a way to now test your hypotheses with things that you can record inside of the brain with animal models. And so it's this bidirectional hypothesis development and testing tool. Now, we've applied the model of HNN to study other signals, including lots of healthy brain signals. We looked at mechanisms of threshold level sensory detection. We've used it to study the origin of spontaneous oscillations in the brain and changes in these oscillations with attention and aging. Most recently, we've applied it to study the impact of non-invasive brain stimulation. You can give electrical current stimulation to the brain and study what is the corresponding result on the neural dynamics. Now, our goal is to make this globally accessible, so we've made it freely available with an online distribution. It's an open source software that's available on this website, hnn.brown.edu. This is the homepage of our website. And you can see there's various menu items, including installation instructions for various platforms. We have tutorials on how to simulate some of the most commonly measured signals, and an under the hood manual that describes the underlying network and some of the theory behind the development of the model, as well as a user forum. The applications of this model have been going on for over a decade, and I show you some examples of that, but it's a brand new software. It came out earlier this summer. We've had a lot of interest, but we're eager to get the community involved and to apply it to the vast data streams that are out there. So with that, I want to thank you and thank my collaborators. This is developed by a multi-institutional team. Two people in particular, in my labs, Sam Nemoynton and Dylan Daniels, have been essential to the software developments and the website developments. Thank you.
Our third speaker is Dima Amso, Associate Professor of Cognitive, Linguistic, and Psychological Sciences. And her work combines measures of behavior, genetics, and brain activity to study attention and memory in infants and children.
Okay, thank you all so much, and thank you to the organizers of this wonderful event. So I'll start by saying that the work I'm going to show is an example of the types of methodological methodological progress that happens when artificial intelligence meets developmental science. So this is Thomas Serre and his team, and these are some of the members of my team, and it's really the collaborative work between the two groups that I'm going to tell you about. So I study human brain and cognitive development. I study the most rapidly changing machine in the universe, which is that human infant to childhood transition. And you know, my goal is to understand not just the development of the human brain and behavior, but also all the environmental variables that shape this developmental course and how that changes across the lifespan. The bottleneck, the issue that we have in developmental science is that human behavior is really complex. And it seems very noisy and very, very difficult to capture. And at the same time, the way the human infant develops into the child is incredibly elegant, and it's very systematic, but capturing that, it has been very elusive. And rather than sort of continue to tell you how systematic it is, but also how complicated it is to capture, I'm going to show you some videos.
Hi, the next case.
So this is a newborn, obviously about a month old, not quite a newborn, and you might think, yeah, there's not much going on up there. But actually, if you pay closer attention, there's a tremendous amount of hand-eye coordination that could be coded and accounted for mathematically, that can be quantified. There's a lot of tongue protrusion. That's actually not all that random. And there's a lot of symmetry in the tongue protrusion. There's a lot of eye movement slash hitting that you see with the hand-eye coordination, and there's symmetry in the arm movements and the legs. And the reason that I know that is that I've had Brown undergrads code and recode these data as part of my developmental lab, and they found a lot of wonderful patterns in that. So not just a few months later, things start to change a lot. You can still see evidence of hand-eye coordination, but now it's changed pretty dramatically. Trunk protrusion is still a behavior. Symmetry in the leg and arm movements is slightly different. There is now babbling, and he looks up. He engages with the person taking the video. He engages socially. And not too long after that, all the same variables that I've mentioned get paired with speech and social engagement that's substantially more sophisticated. So now he's capable of an incredibly difficult coordination task at the same time engaging with a caregiver and moving through space. And this is all happening in a matter of months. And this is him this past weekend, paying it forward, developmentally using all that amazing learning, that hard-earned learning prowess, to teach another baby. So the complexity and richness of this developmental change is really not, again, paralleled in the human lifetime in this first year, 2 years, 3 years. And it's striking and it's rapid. And in fact, we've been really great as a developmental science and neuroscience community at understanding, say, how memory changes, how motor skill changes, but we haven't been really great at understanding how all of these variables come together to form early patterns of change that might be predictive of subsequent outcomes. And again, that's because so much is happening so rapidly in so many systems at the same time. So what if we could quantify this developmental course? What if we could capture that along both behavioral and neural dimensions? And what if we do that over time? Instead of testing the child once, what if we capture the complexity of human development over and over and over again? What if we sample longitudinally? So here I'm showing you, for example, this is hypothetical data, but what if we could do this? Where we've got motor development, attentional development, social emotional development, eye movements, memory, social engagement. I can make cortical development. I can make a list that's endless. And what if we could track this over time in a way that's quantitatively unbiased and in a way that is sort of observant of the complexity of human developmental change? Are there patterns in this course that we could discover very early on that, had we understood them better, would have predicted risk? Is there something here in this very early period that would have predicted the differences that might be observed by the time the child is 10 or 11? And knowing that, could we have done something slightly different in order to sort of steer that developmental course, perhaps even preventative action very early on? So how do we do that? This is, like, as I said, it's been elusive. And so I'm talking to my colleague Thomas Serre, who I've already mentioned is my collaborator on this work. And he has been sort of working in artificial intelligence, computer vision, machine learning, deep network analysis. And he says, well, look, the DNN, the deep neural network analysis of video data, has been revolutionary in a variety of different domains in our society. You know, this is your GPS, being able to track different, stimuli in the visual field. This is what goes on your phone with facial identification. You can play games this way. So what's happening is that there has been this optimization of video data that has made it possible for areas like driving to take advantage of being able to automatically code different object stimuli movements in the environment. What if we could do something like this with capturing the complexity of human development? And that's where the idea for the smart playroom came from. So the smart playroom is right next door. It's just a play space. It looks like a preschool or a classroom. It's not meant to be something that children come into and feel overwhelmed by. It's meant to be quite simplistic. But it's different in that it's wired with a variety of different cameras that allow us to do some of the analyses that I just showed you. We're changing many other domains before. So it's wired with, so this baby is wearing an eye tracker, so you'll see little targets in the next few videos. Those moving targets are exactly where children are looking as they move through the space. That's literally their change in their eye movements. We also put Kinect cameras. Actually, these are Xbox cameras that can help build these 3D representations of the body that can help us track the body through space, and also just track the trajectory of the child as they perform different tasks. At the same time, objects in the room are coded for their identity, for their features, for their color, and for their location in an automatic fashion. The video feeds that we take while children play or engage with a caregiver or do tasks or do memory or attention or motor skill or bounce balls, whatever it is that we're interested in, these video feeds are paired with the tools of computer vision, which is a great application of machine learning. It requires a great deal of early annotation, hand coding, but then ultimately the annotated data are used to develop algorithms that make sense of the image, that learn to code in an automatic, unbiased, rapid fashion per child, per frame, what's going on in that scene. So here's a child playing with one of our experimenters, and what you can see here is that what we're coding for is she happy, is she sad, are her eyes open, is her left eye closed, is her right eye closed, and this is happening as she is interacting. We can do this for both individuals and actually looked at social contingency over time in a completely automatic fashion. We can look at attention and learning and their development in this way. So that little target is the child's eye track as they scan the scene. This is a visual search task. It's a visual attention and learning and memory task. So we can study learning and memory in a very ecologically valid, naturalistic way. We show them an object to look for, just as an example, and the child goes searching. As they do that, there's an automatic coding of all the stimuli as well as whether the child looked at it, so that we understand how are they building a memory of their spatial layout. How does that differ across children, across individuals, across age? And then do they use those fixations that they looked at those stimuli, do they use that as a memory subsequently when they go searching for those stimuli? So that's just one way we can look at attention and memory. We can do much more sophisticated things by, say, building models of the visual scene that children are looking at. So this is a model of literally just the amount of color, flicker, intensity, orientation, and motion information. What that means is that a lot of children, and especially developmentally children, their visual systems don't look like yours and mine. So I'm looking out at you, and some of you are wearing bright colors. That's where I'm going to go. That's because my visual feature detection of color may be particularly sensitive. Children have differences in that, and that developmental change may actually drive looking patterns that are quite different. So we can model that, and then we can look and see what they actually use in the visual environment. We can also look at motor development. So what I'm showing you here in just a moment, she's bouncing a ball, but in a minute we're going to see her skeleton. And what that allows us to do is not only track where she's going, because we can do that in an automatic way, but we can also track, is there a motion for bouncing that we can follow? What does her gait look like? So in cases where development may be atypical with respect to motor patterns, we can track that and detect something subtle, perhaps not available to the naked eye, very early on. And I think what's really critical is that this can all happen at the same time. We're not just looking at memory. We're not just looking at motor development. We're looking at the entire whole complex child. And so you can see that we have these different feeds and different views of the child. This is paired now with cortical data, so we can actually have them wear a new device, a wireless near-infrared spectroscopy device, as they navigate the space, so that we can also get cortical activation data on topics of interest as they do it. We can get heart rate as they do this. We can get galvanic skin response, so if they're getting anxious or not, we can pick that up with changes in sweat, basically. And so this is something that we find incredibly exciting and in a really, really interesting way to start thinking about capturing the complexity and elegance systematically of human brain and cognitive development. So here, the particular excitement is that we're getting the precision of computerized data collection, but with the ecological validity of naturalistic observation. We're getting measures that are pan-cultural, that are not specific to a race or a class. This is child's play. This is children changing from their own baselines over time. It allows us the opportunity to think about putting a smart playroom in Guatemala or a smart playroom in a Syrian refugee camp and actually being able to do the work in a way that we feel is truly faithful to the premise of child development for its own sake, not things that we think they should be doing or we should be doing. And ultimately, I think the most exciting thing is that this allows for discovery. I don't know what I don't know, but if I can look at patterns in existing data and use those patterns to discover something about early human development, I think that's making a lot of progress. And so with that, I'll say thank you.
Do we have questions from the audience? Sir.
Dima, well, very interesting your talk. With this large amount of data, it's not a hypothesis-driven experiment. No? You're trying to discover, as you pointed in the last point of your slide, no? But it's such a rich data that you don't know where to start, no? And this has happened in many places of science. No? When people go to the wild, where to start? What are your thoughts about how are you going to mine this data that is so amazing, so large?
Yeah, this is a great question. So we are taking both a hypothesis-driven approach, so we do actually have very specific questions, and an approach specifically that allows discovery. The hypothesis-driven approach is often in a particular age group, or the discovery approach really allows us to, as we're collecting these data, use classifier analyses to look. If I can bring in a baby at 1 month, and then at 6, and then at 9, and then at 12, and I bring in hundreds of children, and then I call them back and I say, is there anything going on? And we learn that a particular child developed a particular problem. We can go back and look and see whether there was a pattern very early on. So it is discovery, but it's discovery right now at this stage where we have some handle on what the outcomes look like. In the future, if we learn that there are specific patterns in children that, say, might have severe delays with motor development, then that's something that when we notice it in the future is something that we can identify very early on and discuss with the families.
Thank you, great.
Anybody else? So my question listening to, there's a question? Where is it? Sir? Can you go up to the microphone?
I'm sure everybody can hear. I have a question. In some sense, all your projects are... This is for documentation. All your projects are in some sense small data projects in the sense that the number of subjects is relatively small, and I just wondered whether you would each talk a little bit about what your thinking is if one had big data in the sense of large numbers of subjects that are quite varied. So, I mean, even in Professor Harrison's case, I wondered how much variation there was in even those brain signatures for a given action. But imagine that one had a sample of 1,000 subjects. There's bound to be a lot of variation. I just wonder how you all think about that and whether you think that would make things enormously more difficult or it would rather confirm the patterns you think you see with small amounts of data.
Do you want to start, Matthew?
Yeah, that's a great question. So in the brain-computer interface technology, when they put an electrode in the brain, they know sort of generally what region they're putting it in, but they have no idea sort of exactly what neurons the sensors are going to pick up. It's not even clear that that makes sense because every human's brain is likely very different, particularly at the level of individual neurons. And even from day to day, that sensor, even though it's very tiny, is very large relative to a brain cell. And it might wiggle around or move, or maybe the brain changes. So this calibration that you saw, where the sort of algorithm has to figure out what the brain is, this correlation between brain and behavior, or brain and intention, that has to happen again and again. And I think, so there's definitely, I don't think that there's the possibility that with, let's say, thousands of BrainGate patients, that you're going to ever get to the point where you can just, plop in a sensor, and based on all the other subjects, it will know automatically, what this person is thinking. There's going to have to be some sort of calibration phase. But I do think that big data in the way that you're talking about, sort of many subjects, does have the potential to improve this technology, because there's likely lots of statistical regularities, even though everybody's brain is different, that can sort of be, that can facilitate this process of calibration. For instance, understanding how variable neurons are, or just trying to identify what are the features that really matter when you're looking at this brain data. And mathematically, that might show up in things like choosing the widths or the shapes of different kernels. That you somehow you can learn something from different patients to improve one. But right now, the sort of technology is not there. I will also say that this does feel like big data, even though there's only one subject, right? Because a single participant is generating roughly a terabyte a day, and so there's a lot to get through and mine.
So yes, thank you for the question. It's a great question. I'll say a couple things in regard to EEG. One is, because it's so easy to record and because we now have open science, there's a tremendous amount of data out there that's available for everybody to look at and to start to analyze. And the other thing about EEG, which is often viewed as a downside, but I see it as a plus side, is that it's called this macro scale signal, and like large ensembles of neurons need to be doing the same exact thing in order to record it outside of the head. Now, because of that, there are limited signals that we record outside of the head. We typically see things like ERPs, event-related potentials, or low-frequency oscillations in very stereotypical frequency bands. And so there's a lot of consistency across subjects. We see similar patterns of activity in a subject that comes in 1 day or 4 weeks later. And so that gives us really some targeted points that we have confidence that we'll see even in very large data sets, some commonalities, and we can apply these artificial intelligence algorithms to them and classify what are the different states. The other thing is that it's really dictated by the structure of the neocortex, this outer layer of the brain, which has very characteristic features throughout the brain and across brain areas and across species. And so that also limits the space of signals that we have to work with outside of the head, because there's only so much you get from the structure of the cortex. Nonetheless, there's very meaningful information, as you saw in the autistic example, that there are vast differences in these disease states and across attentional states and such.
We are actually moving towards, we wanted to develop this space and are in sort of now testing in the hundreds, but ultimately we would like to be testing in the thousands. To get at that individual variability. We think it's important. It's not noise, it's signal. So.
Hi, thank you for your presentations. In terms of Professor Harrison's models, it seemed like the deep neural net or whatever algorithm you're using, it had to know that it was deciding which direction to move the cursor. With the smart playroom, it seemed like you had very specific features that you were trying to track, such as right eye movement, left eye movement, where the focus of the child was. So I'm wondering, where does the contextual subject expertise end, and where does the machine learning begin?
So in the video that I showed, where the person was playing the little dot game, so let me start over. So the type of machine learning that we were using there needs, it would be, for those of you who are familiar with this, it would be called supervised. It needs brain data movement pairs. in order to build up that mapping between brain data and movement. This would be the training data. And of course, the person is paralyzed, they can't move, and so the sort of clever innovation that BrainGate sort of came up with is to just ask them to pretend to play a game and just hope that they're trying to move the cursor in the direction. And so that gives you this sort of second variable, the movement intention. And so, so you collect those pairs in a supervised way during this sort of calibration phase as you're basically collecting training data. And now the video that you saw, it was simultaneously collecting training data and improving its prediction using sort of Bayesian techniques so that it can sort of incorporate data on the fly. But eventually the training period ends, and then the dictionary is fixed, and the algorithm is just always predicting what the next movement is. And it's not really collecting new training data, although they're thinking about ways to do that, sort of looking back in the data and saying, oh, this person went to the letter A, and that's probably what they wanted to do, and so we could get more training data. I don't know if does that answer your question, or?
I was more wondering, so like if you put that chip in a different part of the brain, it would be collecting totally different signals. Would the model know that it's not looking at the correct signals anymore?
Yeah, so the question was, if that chip was put into a different part of the brain, would the model know that it's not looking at the right part of the, something different? And the answer is it depends. If it was still in this particular, the algorithm that I showed, if it was still in the calibration phase, it would begin collecting new dictionary data. And if it collected enough of it, it would sort of discount what it had learned before. If it was already in the I'm going to use it phase, and you just took the chip out and moved it, it just wouldn't work. It would be seeing brain data. It would think it was coming from, it meant one thing. It's like switching from English to German or something. It just wouldn't understand what to do. Now, it's possible that the participant is also a learning machine, that perhaps they could eventually adapt and be able to use this technology, maybe, but it would be much longer and sort of much more, they wouldn't be thinking, I think, so intuitively about moving. And maybe it wouldn't even work at all. Is that a better answer? Yeah. Actually, I'm the only one that needs this, right?
So is this on? With the smart playroom right now, the distinction where sort of the human experimenter ends and where machine learning begins is we still are identifying the behaviors that then get hand annotated in the video feed, and then those hand annotations are used to train the machine learning algorithms to be able to detect those patterns in now completely novel data sets, so that you can do that in a way that's sort of quantitative and unbiased and across a variety of different patterns and behaviors.
Anybody else? So I guess if we have no more questions, we can move on to the reception portion of the evening. Is that what you're thinking? Okay. Okay. Thank you very much.